Chromosomal deletions. Larger deletions of the chromosomes, they are not compatible with the life. 10 to 15% are the chromosomal deletions which are translocations, while remaining 85 to 90% are the true deletions of the chromosomes. In this diagram, we can see that how chromosomes are deleted. If we do a comparison, that a small portion on the tip of the chromosome has been deleted. If there is deletion on the chromosomes, that the individual contain such chromosomes which have been deleted, they usually do not survive. Chromosomal deletion. In this diagram, we try to understand that how the portion of the chromosomes are deleted. This is the structure of a complete chromosome, and within this chromosome there is a fragment that is represented as C and D. This fragment has been deleted from the chromosome. So this fragment can consist of a single gene and it consists of many genes. Chromosomal deletions are of two types. One is called as terminal deletion or terminal deficiency, and second one is called as interstitial deficiency or interstitial deletion. When a small fragment of chromosome is broken from the end of the chromosome, then it is called as terminal deficiency or terminal deletion. Here we can see that a small fragment at the tip of the chromosome has been deleted. While in case of interstitial deficiency, the fragment which is present in the middle of the chromosome will be deleted. This is the fragment that consists of, that is marked as 2 and 3. If this fragment which is present in the middle of the chromosome, if this fragment will be deleted, then it will be called as interstitial deficiency or interstitial deletion. So chromosomal deletions are major chromosomal deletions are of two types: terminal deletions and interstitial deletions. Phenotypic consequences of the deletions. It depends on the size of the deletion, that how much size of the chromosome has been deleted, or it also depends on those genes which were present in that particular fragment of the chromosome. Usually, the deleted portions, if they have important genes, then due to the deletion part, the consequences of the deletion are detrimental or threatening to the life. In this diagram, we can see that how fragments are deleted within the chromosome. If we do the comparison between the two chromosomes, this fragment, that is larger one, and from here a small portion of the chromosome has been deleted. If we do the comparison between the dark bands and light band, these are the dark band, second dark band and third dark band. But here there is a large portion of chromosome, while some of the part of this chromosome has been deleted here. So this is the deletion which is called as interstitial deletion. Interstitial deletion which has been identified at chromosome 16. If we see here chromosome 16, in case of chromosome 16, one of the chromosome is larger while the second chromosome is smaller in size. So some part of the chromosome has been deleted from this point. So this is also the type of interstitial deletion. Terminal deletion. Terminal deletion in case of humans have been seen in a syndrome. The name of syndrome is Cry-do-Chat syndrome. In this case, usually a small fragment of the chromosome is deleted from the tip of the chromosome. So this portion of the chromosome has been deleted. If we do the comparison, here is the larger portion of chromosome and here is the smaller portion. So a small portion of the chromosome from the tip has been deleted. Terminal deletion in case of chromosome 6. Another example of that chromosomal deletion, we can see chromosome 6. In case of chromosome 6, here is the small portion of chromosome. If we magnify chromosome 6 here, that this chromosome is larger one, while its homologous chromosome is a smaller one. A small portion from the tip of chromosome 6 has been deleted. So there are two major types of the deletion in chromosomes: terminal deletion and interstitial deletion. In addition to these two major deletions, some are micro deletions that can be in case of few bases.